You look at a piece of moldy bread or a smear of bacteria on a lab dish, and you assume you're looking at something as close to mindless as biology gets. No brain, no nerves, no decisions, just chemistry unfolding on autopilot. A colony of cells doing whatever their DNA hardwired them to do, with about as much agency as a rock rolling downhill. Now consider this. A single bacterium sitting alone in a drop of water will often do almost nothing, quietly waiting. Get enough of them together, past a specific threshold, and the entire population can switch behavior in unison all at once, as if something had just been decided. The question of whether that counts as a decision turns out to be far less obvious than it sounds. Start with the discovery that broke open this entire field. A research team at Princeton spent years studying a glowing marine bacterium called Vibrio fischeri, which lives inside the light organ of the Hawaiian bobtail squid. The squid uses the bacteria's glow at night to erase its own shadow in moonlight, hiding it from predators lurking below in a defense mechanism called counterlumination, matching the faint moonlight filtering down from above so precisely that a predator looking up from beneath sees no silhouette at all. Here's the strange part. A small number of these bacteria, isolated or thinly spread, produce essentially no light at all. But once their population inside the squid's light organ crosses a specific density, the entire colony begins glowing in near-perfect unison, as if a switch had been flipped everywhere at once. The Princeton team's research revealed how that switch actually works. Each bacterium constantly releases a small signaling molecule called an autoinducer into its surroundings. In a sparse population, that molecule drifts away and dilutes into nothing, essentially invisible. But as the population grows denser, the concentration of the molecule in the shared environment climbs along with it. And once it crosses a specific threshold, individual bacteria begin detecting it in large enough quantities to trigger a genetic switch, turning on the genes responsible for producing light. The same research group went on to identify similar signaling systems across a huge range of bacterial species, and found that many species produce not just one autoinducer molecule, but several different ones simultaneously, effectively running multiple parallel head counts at once, some tuned to detect only their own species, and others tuned to detect any bacteria in the vicinity regardless of species, giving colonies a way to gauge both how many of us are present and how many of everyone are present in a shared environment. This process, now called quorum sensing, is essentially bacteria counting their own numbers using chemistry instead of census, and only acting once they've confirmed there are enough of them present for the behavior to actually matter. The word researchers borrowed for this is telling. A quorum, in human terms, is the minimum number of members needed before a group is allowed to vote or take official action. The same concept that keeps a city council or a corporate board from passing a binding decision with only 2 people in the room. Bacteria, it turns out, run something functionally similar. A handful of Vibrio fischeri glowing alone would waste energy and accomplish nothing, since the light would be too dim to hide the squid's shadow anyway. Waiting until the population is large enough for the light to actually be useful is, in a very literal sense, the more efficient strategy, and quorum sensing is the mechanism that lets a population without any central brain, without a chairperson calling for a vote, without anything resembling deliberation, somehow arrive at that same efficient outcome anyway. This same mechanism turns out to explain something far more dangerous than a glowing squid. Many pathogenic bacteria use quorum sensing to time their attacks. A small number of invading bacteria releasing toxins immediately upon entering a host would trigger an early immune response and likely get wiped out before accomplishing anything. Research on bacterial virulence has found that many pathogens instead stay quiet during the early stages of infection, multiplying without releasing harmful toxins, essentially hiding in plain sight while their population count climbs. This delay can last hours or even days, long enough for the invading population to grow from a handful of cells too small to matter into a force large enough to matter enormously, all while the host's immune system detects little to nothing worth responding to. Only once quorum sensing signals confirm the population has grown large enough to overwhelm the host defenses do the bacteria collectively switch on toxin production, all at once, in a coordinated strike that individual, isolated bacteria could never have pulled off, and that no bacterium ever needed to individually decide to launch on its own. Understanding this timing mechanism has become a genuine target for new kinds of antibiotics. Ones designed not to kill bacteria outright, but to jam their ability to sense a quorum in the first place, keeping a dangerous population permanently convinced it's still too small to safely attack. If that sounds like a form of group-level strategy, the next discovery pushes the comparison even further, directly into territory usually reserved for brains. A research team at the University of California, San Diego, discovered that bacteria living together in dense communities called biofilms communicate using electrical signals remarkably similar to the ones neurons use to fire messages across your brain. Their research found that bacteria in the interior of a biofilm, cut off from an easy nutrient supply, can send electrical pulses outward through the colony by passing potassium ions from cell to cell, the exact same basic mechanism neurons use to transmit electrical signals along a nerve. Using fluorescent dyes that light up in response to changing potassium levels, the research team was able to literally watch waves of electrical activity ripple outward across a biofilm in real time, radiating from the starving interior toward the well-fed outer edge, a visual that looked startlingly similar to footage of electrical activity spreading across neural tissue. When outer bacteria detect these pulses, they respond by temporarily slowing their own nutrient consumption, effectively rationing resources so the starving cells in the interior can survive. In further experiments, the same team found they could even trigger these electrical waves artificially and use them to synchronize the behavior of separate, physically distant biofilm colonies, essentially getting one community of bacteria to influence another purely through electrical signaling across the shared environment between them. The colony, in other words, appears to be relaying information about a local crisis and adjusting collective behavior in response, using a signaling method that, at the level of basic mechanism, isn't so different from what happens inside your own skull when one region of your brain needs another region to change what it's doing. None of this means bacteria are thinking in any sense resembling human thought. There's no evidence of anything like awareness, no experience of choosing one option over another, no internal deliberation weighing costs and benefits the way a brain does. What quorum sensing and biofilm signaling demonstrate instead is that sophisticated, coordinated, population-level behavior doesn't actually require a brain to produce it. It can emerge from nothing more than simple rules, chemical thresholds, and electrical relays distributed across thousands or millions of individually mindless cells, each one following a basic local rule with no awareness of the larger pattern its behavior is contributing to. That distinction matters because it reframes the original question. Bacteria almost certainly aren't deciding anything in the way you decide what to eat for dinner. But the outcome produced by quorum sensing looks, from the outside, remarkably like a decision. Information gathered, a threshold evaluated, a coordinated response triggered only once the evidence supports it. The mechanism underneath is nothing like a brain. The behavior it produces, at a population level, can be difficult to tell apart from one. Even single, isolated bacteria show something that looks uncomfortably close to decision-making, without any group coordination involved at all. A Harvard research group spent decades studying how the bacterium, E. coli, moves through its environment and found that individual cells navigate using a pattern now called run and tumble. A bacterium swims in a fairly straight line, powered by a spinning tail-like flagellum, for a short stretch, then abruptly tumbles in place, randomly reorienting itself in a new direction before swimming straight again. On its own, that sounds like nothing more than a random walk. But that research found the randomness wasn't uniform. When a bacterium happened to be moving toward a higher concentration of a useful chemical, like a nutrient, it would suppress its tumbling and keep swimming straight for longer than usual. When it was moving away from that gradient, it would tumble sooner cutting the unhelpful direction short and trying again. The cell has no eyes, no memory of a map, and no way to sense the overall shape of the gradient at once. It's comparing the concentration it detects right now against the concentration it detected a few moments ago, and biasing a fundamentally random process based on whether things are getting better or worse. That's not deliberation in any conscious sense, but it is, in a very stripped-down way, a running evaluation of outcomes feeding back into future behavior, which is most of what a decision actually requires functionally, even without anything resembling a mind behind it. So the next time you glance at a colony of bacteria multiplying quietly in a petri dish, or notice mold spreading in a slow, patient bloom across a piece of forgotten bread, consider that somewhere in that unremarkable smear, trillions of cells with no brain between them are constantly counting their own numbers, comparing their surroundings moment to moment, and preparing to act in perfect, brainless unison the instant a threshold is crossed.